Hello, and welcome to the KCSA Mental Health Virtual Investor Conference. My name is Allison Sauce, and on behalf of KCSA Strategic Communications, as well as our co-host, the OTC Markets Group, we're very pleased you have joined us for our next live presentation from PsychoGenics. Before I introduce our speaker, please note you can submit your questions for the presenter in the question box to the left of the slides. Additionally, all of today's presentations will be recorded and available for 24/7 replay. At this point, I am very pleased to welcome Daniela Brunner, Chief Innovation Officer of PsychoGenics, a private company. Welcome, Daniela.
Hi, thank you for the introduction. Thank you for attending this presentation. It's a bit long, so in case we don't have time for Q&A, please do submit your questions to our CEO, Imer Lihi. I will present this contact at the end of the talk as well. So today I would like to talk about how we do drug discovery. We do drug discovery internally and with our partners. We have three branches of our activity: the partner pipeline, our CRO, and our proprietary drug discovery pipeline. The CRO gives service to our partners, our clients, and our internal drug discovery. And everything centers around our high throughput drug discovery platform that I will explain in a minute. So we focus on mental disorders, CNS disorders. Probably I don't have to say much about these. 1 in 5 adults experience some kind of mental illness during their lifetime, with 1 in 20 having a very serious mental illness. So it's an important unmet need, and we hope to bring novel therapies, novel mechanism of action to the market. So why is it so difficult? This is not a one target indication. It's not as simple as say, I have a target, I have a mechanism of action, and if I have a drug that can tackle that, then I have the road to the clinic clear. In fact, CNS disorders are very complex. There are lots of epigenetic factors and susceptibility genes, and all these interact with each other and with the environment. So we believe that a phenotypic drug discovery engine, which is based on in vivo efficacy, is the way to find novel drugs which are efficacious, and also to make the whole pipeline of drug discovery much more efficient and cheaper. So I hope I can explain how we do this. The key of what we do is this AI-enabled drug discovery platform. In fact, we have 4 different platforms: SmartCube, NeuroCube, PhenoCube, and eCube. The main screening platform is SmartCube, where we can screen drugs very quickly. 3 of these, SmartCube, NeuroCube, PhenoCube, use computer vision. It's a type of machine learning where we can go over videos and very quickly extract information of what our subjects are doing and how the drugs are affecting their behavior. This is the key component of our in vivo phenotypic platform. We also use eCube. This is based on EEG. This is very good as a second tier to find biomarkers that could be very important in the clinic. And we then mine all the data using machine learning AI algorithms to define signatures of drugs or animal models of disease, or to answer any number of other questions in a way that I will present to you in a minute. These platforms are protected by a strong portfolio of patents. And as I said, the most important one for screening is SmartCube system. SmartCube uses 3 different types of cameras and mechanical actuators to follow mice around and capture everything they do. So everything that can be captured is captured by this system. The hardware is of our design, And the way the animals are running this platform is always standardized. So we have been running the same protocol for many, many years. And the beauty of that is that then the data is very comparable to each other. So we can run reference compounds, and then 3 years later, we can use that data to compare against novel compounds. I will show you how we do this. So these proprietary hardware, proprietary computer vision, and machine learning analysis tools allow us to do very standardized analysis of drugs. This is a one session, so it's in one session of 45 minutes. We can capture a lot of information about any drug. If the PK is appropriate, if there is an efficacious signature, what kind of signature, is this a good antidepressant, antipsychotic, anxiolytic? This is all done in one session. And this is the key of how fast this system is for drug discovery and for SAR. So how do we use a system? We first run lots of reference compounds. We have hundreds of reference compounds already run at many different doses, 2, 3, 4 different doses. So with the characterization of these reference compounds, we can now proceed and create these machine learning classifiers that learn from the reference compounds how an antidepressant looks in this system, in a mouse, in the SmartCube system. And then anything that looks like it will also be called an antidepressant. So we train machine learning classifiers to recognize 14 different classes of potential therapeutic effects. With this in hand, we can run novel compounds, and then we present the novel compound to the classifier, and then we basically diagnose the novel compound and see what is the therapeutic efficacy that is predicted by this system. The system is very fast, so we can run up to 9,000 compounds, different doses in a year. We can run full libraries with this system. So here you can see an example of a class analysis at the top left, subclass analysis, which is closer to mechanism of action. And then on the right, this is what we call similarity analysis. So let me explain a little bit. The green color is an antidepressant signature. So this compound, in this case, is a mGlu2/3 antagonist, which is a novel mechanism that the classifier didn't know. This compound comes out as an antidepressant. That's a green color on the class analysis. On the subclass analysis, the mechanism of action, it's novel because it's not in the classifier, but it's most similar to an SSRI and to a tricyclic mechanism of action. On the right, you can see the spider plot shows the similarity of this compound against different reference compounds. This is what we call the psychedelic panel. It has going clockwise psilocybin, psilocin, and then other compounds, LSD, ketamine, the enantiomers of ketamine, and a number of negative controls. This is a very granular analysis that says, well, this drug may be an antidepressant, but what kind of antidepressant? If it's close to a psilocybin or psilocin, then of course it becomes interesting. It may be a psychedelic-like compound in their phenotypic action, not necessarily a compound that will cause hallucination, because we can always check that the mechanism of action is not causing hallucinations, and we can follow up in other assays. So this allows us to take a compound or a library of compound and go very quickly and find interesting hits that can be then used to develop a novel candidate. So this system can be used, as I said, for screening of libraries. We can also screen compounds that are already in the market and find a new indication. We can also turn it into a target-based screening. There is no reason why not to do that if there is a target of interest. We can test combination of compounds and look for synergy or potentiation, and then it's used as for lead optimization when we need to fix maybe the PK or other aspects of the drug. Because we are testing efficacy, toxicity, brain-blood penetration very early on in the drug discovery pipeline, the system is much, much faster. So here is a quick comparison between the number of compounds needed to reach a candidate, with an industry average of more than 2,000 compounds needed to be synthesized. Anything in blue are compounds that have been discovered using SmartQ. These are in partnership with the different companies, Sunovion, Roche, Blue Oak. We also have a partnership with Karuna, which I'm going to show you in a minute. So in any case, the point here is that because of the way the system works, we can get up to a candidate in much faster in terms of synthesis and time, and therefore the cost is much, much lower than the typical drug screening pipelines in industry. So going to partner drug discovery, which is one of our three branches, we have, as I said, some collaboration with Sunovion, Roche, Blue Oak, and Karuna. The collaboration with Sunovion, we have a number of compounds in the clinic. The more advanced is A56 or ulotaront, which is in the phase 1, 2, and 3 for schizophrenia, major depressive disorder, Parkinson's disease, psychosis, and bipolar disorder, and agitation in Alzheimer's. For ulotaront, we are in the phase 3. We're going to have readouts early next year, and it's a very exciting novel mechanism of action antipsychotic. So ulotaront was discovered using SmartQ. In here, you see a comparison with haloperidol and clozapine. Haloperidol being a typical antipsychotic. The system uses the purple color to give you a sense of the indication. So light purple is a clean antipsychotic dose. Haloperidol very quickly goes into this dark purple color from 0.1 milligram per kilogram to 0.3. That is a dose that will produce very strong motor side effects. Similarly, clozapine between 1 and 1.5 milligram per kilogram goes for a clean antipsychotic to having some side effects. In contrast, ulotaront from a dose of 3 all the way to 30 milligrams per kilogram shows a very clean antipsychotic indication, and this was something we replicated several times, and was exciting. It was confirmed with other secondary essays, and so we took it all the way to the clinic. This was led by our partner Sunovion. The phase 2 results were very interesting and very positive. We had very positive in total symptoms in the PANSS scale, but also looking at the negative symptoms, we had a very strong effect. Interestingly, in a follow-up open label study, the symptoms kept improving, as you can see on the right, all the way up to 7 months of treatment. The ulotaront is a TAR1 5-HT1A agonist, so it does not hit the D2 receptor at all, and that's a big difference with the vast majority of the antipsychotics now in the clinic. The side effect profile was very favorable. Moreover, for insomnia and anxiety, there were fewer side effects, suggesting a very broad efficacy profile for ulotaront. So for our internal drug discovery, we also use the proprietary pipeline. We have a library of active compounds that we can scan and look for interesting hits. And I'm going to show you some of the data that we have obtained from this library. Our internal drug discovery, it's a very strong group of 10 drug hunters, which have a combined years of experience, which are above 200 years of combined experience. We use the internal drug discovery, as I showed you before. We have a library of more than 5,000 active compounds, which are chemically diverse, and we continue to add new compounds to our library using machine learning techniques. And we have right now compounds for agitation, depression, and schizophrenia. I will show you the pipeline in a minute. This is an overview of the library right now. As before, we use colors to quickly provide a very quick readout of the efficacy. So everything in every line here is a compound at a particular dose. Anything green is a potential antidepressant, purple antipsychotic, in yellow anxiolytics, and then we also have this gray color, which indicates compounds that are very active but do not resemble anything in our current database. What is very interesting is that because we're looking at the phenotypic effects of the drug downstream from their action as multiple targets, a very small change in the ratio of activity at different targets may change the therapeutic effects. For example, on the left, you see a compound with a strong yellow signature, which is our anxiolytic color. By small changes in the pharmacophore, we have seen these over and over. We can turn these anxiolytic into antidepressant or into an antipsychotic. So we have pharmacophores that keep giving very interesting compound, and this is what's called the privileged structure, where you can have a whole series of analogs derived from an initial hit and then have different indications for the different analogs. Our internal drug discovery has produced very interesting compounds. We are entering IND talks now for agitation and aggression, next year for treatment-resistant depression, and we have a number of other compounds, some compounds for treatment-resistant depression, which are based on similarity to psychedelics, but without the dissociative aspect. And we're also working on pain, epilepsy, and later on Parkinson's. And this is one example of our internal drug discovery. As I said, these are we're looking for psychedelic-like compounds, but they do not have a dissociative aspect. So what we do is we look at phenotypic similarity to compounds in our database, in particular psilocin, psilocybin. We call those psilomimics, or similarity to ketamine. We call them ketamimics, or similarity to MDMA, and we call them molymimics. So we call them mimics because they are similar at the phenotypic level, but not at the mechanism of action. So how we do this, we comb all our libraries and we look for similarity to, as I say, psilocin, psilocybin, ketamine, and molly. And here you can see the spider plots that I explained before. The first two on the left are psilomimics, and you can see a maximal similarity. This is this purple color to psilocin and psilocybin. The ketamimics, the first one looks like S-ketamine and the second like R-ketamine, so we can look out for similarity to particular enantiomers. And then molymimics this is the green dot showing maximal similarity to molymimics. Once we find these hits, then we start a project around them, and the project follows a standardized pipeline from SmartCube. We look at similarity to these compounds, and then we go into in vitro pharmacology because we want to exclude anything that hits the 5-HT2A, 2B, and MDA, and other targets. So the anti-target panel allows us to focus on novel pharmacology. To make sure we are not looking at anything that has a dissociative aspect, we are going to use the head twitch assay and locomotion to make sure that our compounds are not sedative. We also look at pre-pulse inhibition of startle. That's a way to, in mouse or rats, to differentiate against some of these psychedelics from the literature. We use a forcing test in rat or mouse. We use a special version of it that's sensitive to the longer terms effects of ketamine and hopefully other treatment-resistant depression candidates. Social defeat is another way to test our candidates. And then, of course, we have some preliminary toxicology to define the lead series. This is our very typical table showing all the data we gather for each of the compounds. At the top, you see the different signatures of the compounds. The green color be the antidepressant. Some of these antidepressants are very stimulant, and that's the blue color. So we can do very quickly synthesize novel analogs, run them in SmartCube, make sure we still have the signatures we like, make sure we still have a similarity to different reference drugs we like. And then, anything in green here, these are, in vitro assays looking good. Basically, that's where we are. We define our series and our lead candidates. This is, again, just one example of one of our projects. And this is my last slide. So we have a little time for questions. So if we don't have, we have one question coming from the public. The question is, our company is private right now, and we are looking into either venture capital investments or possibly going public. Another question is if our partnerships are producing revenue. We expect that the revenue will come very quickly because we have our most advanced drug is in phase 3. We expect positive results next year. And so, possibly in 2 years. I mean, every partnership is organized in a different way. There could be milestone or royalties. We are pretty flexible in that respect. And so eventually, yes, the partnerships will be producing revenue. So regarding another question about our IP portfolio, So we have a very strong set of, we have all the chemistry is protected by IP. So we have a number of patents filed or being filed protecting all our internal drug discovery. The partner drug discovery of course is also protected by IP, and our technology is also protected by IP. The other thing is that we've been doing this. I mean, right now there's a lot of hype about AI and machine learning. We designed SmartCube 19 years ago when nobody was speaking about machine learning for behavior. Maybe there was one academic lab that mentioned using machine learning for behavior. So we turned this drug screening platform, and we've been using it for maybe 15 years. So it was designed, it was validated in a couple of years, and we started using it, and it's been uh, quite amazing, I have to say. After developing SmartCube, we developed the other cubes. But it's a platform, as I say, we're in phase three. I don't know of any other truly machine learning development platform that has produced so many drugs right now in clinical trials. So I think that's possibly the end of the time, with a little bit more time. But as I said before, if you want to contact us and have follow-up discussions, please contact our CEO and president, Email Lihi, and here you have her contact number, email. So thank you so much for your attention.